Today I'm going to talk to you about a new method for cell culturing that has enabled the scientific community to accurately study in vivo human cellular systems. These advances in experimental models are very interesting to me because I perform cell culture-based experiments as an undergraduate researcher in a pancreatic cancer lab. 2D culture has been the accepted method for growing and studying cells for the last 100 years and has provided important information about cell biology over that time. This method consists of growing cells on a flat surface, the bottom of a petri dish or a flask, surrounded with media within the dish. According to a study by Duval et al., published in 2017 in Physiology, the monolayer of cells have access to the same amount of growth supplements and nutrients within the cell media, which allows for evenly distributed growth and proliferation of the cells. Due to this, 2D culture is very useful for fast, cost-effective cell experiments. On the other hand, 2D culture is not the best model of cell environments inside the body. Inside the body, cells are surrounded by different cell populations, extracellular matrices, and other stimuli that 2D culture is not able to provide. In 2020, Frontiers in Molecular Biosciences published an article by Jensen and Tang, in which they explained that these cellular interactions with the environment have a large impact on many aspects of cell function, including differentiation, gene expression, and mobility. This makes it very clear that 2D culture, due to the lack of proper cellular environments, would not represent in vivo systems, and the cells would react differently in 2D than they would if they were in the body. Another widely used experimental system are animal models. In 2015, Future Science published an article titled "Animal Models Are Essential to Biological Research," in which the authors claim that the vast commonalities in the biology between mammals, along with the fact that human diseases are present in other animal species, makes animal models essential to studying human biology. Animal models are also able to show the effects of whole organism, whole body systems on cellular functions or diseases, while 2D culture systems cannot. The main downsides to animal models, and why it is not a viable option to study human in vitro biology, is that there are genetic differences between animals and humans, and so the results of animal model studies are not always translatable to humans. Both 2D culture methods and in vivo animal studies fall short of being accurate models for human in vivo studies. While both have provided valuable knowledge of biological systems, new methods are needed if we still want to study human biology. A relatively new method of cell culture, 3D culture, has fulfilled this need. 3D culture allows cells to interact with their environment in 3 dimensions, just as cells do within the body. This culture method also allows for the environment of the cells in the culture to be customized to mimic specific cells or tissues found within the body. According to an article published in 2018 in Frontiers in Pharmacology, the customized cell environment of 3D culture includes cell-to-cell and cell-to-matrix interactions, nutrient and metabolic waste, and other tissue-specific scaffolding cells. Cells harvested from patients are placed in 3D culture. They are able to retain their in vivo characteristics and morphology, as shown in a study by Yamada and Kuckerman in 2007. 3D culture has been proven to be a more accurate representation of in vivo environments so that cells can be studied as they are in vivo, in real time, while being outside of the body. 3D culture allows for accurate drug treatment and development studies, patient-specific research studies, or organ biogenesis, and the investigation into both healthy and diseased tissue states to answer fundamental biological questions. This model can accurately mimic how cells react to drug treatments inside the body, which provides opportunities for more efficient drug testing and human applications. It then becomes possible to use 3D culture for patient-specific therapeutic testing, as samples taken directly from the patients can be grown in 3D culture and tested to identify targets and mechanisms which can then be applied to the patients themselves for individualized treatments. Patient cell samples grown outside the body in 3D culture can retain their in vivo characteristics and function, so that these cells can be used to grow into replacement organs or tissues derived directly from the patient. This culture method provides dynamic and instantaneous analysis of in vivo cellular systems that can be changed to either healthy or disease states based on the environmental cues provided, allowing for the gain of knowledge of many biological systems and diseases in development. The use of 3D culture will provide answers to fundamental biology questions and cellular function that would have otherwise been impossible to study outside of the body. I hope this presentation has given you an exciting insight into new experimental models that are paving the way for advancement in biological understanding. Due to this new method, we can only imagine what the next big biological breakthrough will be. Thank you.